The overall goal of this procedure is to isolate PCR-ready nucleic acids and immunoreactive protein from a sample without the need for electricity or laboratory infrastructure. Isolation of nucleic acids and proteins begins with mixing the sample with guanidine thiocyanate to lyse the sample and passing the mixture over the sorbent in the extraction pipette 5 times. After the 5th and final passage over the sorbent, place the mixture into protein extraction buffer and set aside for isolating the immunoreactive protein. Next, the sorbent and bound nucleic acids are washed by passing 95% ethanol over the sorbent numerous times. The sorbent and bound nucleic acids are then dried by passing ambient air over the sorbent for 5 minutes. The final step for PCR-ready nucleic acid isolation is to elute the nucleic acids from the sorbent using 250 microliters of tris or another similar aqueous solution. For isolation of the immunoreactive protein, after mixing the sample and protein extraction buffer, use a second extraction pipette and pass the mixture over the sorbent 15 times. The next step is to wash the sorbent with bound protein by passing 95% ethanol over the sorbent numerous times. Then, the sorbent and bound protein are dried by passing ambient air over the sorbent for 5 minutes. The final step of the protocol is to elute the protein from the sorbent. Ultimately, the extraction pipette used in this procedure allows for the electricity-free isolation of PCR-ready nucleic acids and